So we're going to have Romain Guyon from the University of Oxford that is going to talk to us about his research on microcapsule for single-shot vaccination. But first of all, we have Stefan Jungbluth that is going to talk to us about his career from dopamine to vaccine, or what is it that you do at work, son? So I'm going to do just a small introduction, but I will not say too much because otherwise I will do too many spoilers. So Stefan has been in the AVI since June 2012. He first joined as a business manager and now he's a director of external affairs. He was previously co-founder of a biotech in Spain focused on discovering and developing drugs for rare cancers. And originally, he has a PhD from the LMU and an MBA from IE Madrid. So, Stefan, I will let you share your screen, and thank you very much for being here. We are happy to hear about your story.
Yes, Allegra, thank you very much. So let me see if I get this right here. Can you see now the full screen?
Yes, perfect.
So, yes, so great.
Sorry, one last thing. If you have any question, please write them on the Q&A below. As soon as the presentation finish, we will ask Stefan a couple of burning questions, and then at the end of the two talks, we will do a Q&A session. Sorry, Stefan, go ahead.
Yes. So, yes, thanks again, Allegra. So, this is, as she said, more in my career. So, hope this may serve as a stimulation or or the opposite for your own career. So, and when I was asked to give this presentation, this reminded me of how difficult it is to explain to people what you are doing. And I think this gets the more you advance, at least in my case, the more I advance in my career, the more difficult it got to explain what I'm doing. And the moments when I realize this, when I get reminded of this, when you have to fill in forms. Sometimes you have to fill in forms for visa applications, for example. Then they ask occupation. And I've never filled in any of these forms, whereas I saw biologist, researcher, anything. There was never ever anything near to what I was doing at the time. There's always the other profession options. But so to give you an example, this is from the Chinese visa application, which you had to fill in a couple of times. And so you can see here again, as usual, nothing anywhere to what I'm doing. But this is an interesting example. If you look at this box here to the right, you click any of these options, and I think you directly disappear from passport control to interrogation center somewhere in China. And on the left, there's a more real life options, I think, but even there, nothing fits me. So also here, I had to go for the other option. Maybe next time I try the entertainer box here on the left to see what happens when I check this. But so this shows you, me at least, how difficult it is. I never fit any of these predefined categories. And this is also why I gave this title to my presentation. I've failed, I have to admit, miserably when I tried to explain my parents what I was doing. They repeatedly asked me, "So Stefan, what are you doing actually?" And I tried, but I have to say I failed. So this presentation is a kind of an act of redemption, if you want, for this previous failure. So this is chronologically, I think this is probably the best to explain a little bit. So how my career went. Not everything is necessarily very exciting for you here, but just for the sake of completeness, let's say. And I make some excursions then into my current job and on a year and a half I spent at the European Commission. So I'm by training a biologist. I studied it at Bonn University in Germany with a focus on genetics and biochemistry. So you see, it's quite a while ago. This was an exciting time, I have to say, end of the 80s when really the molecular biology took off, genetics, PCR was invented. So it was pioneering times then. And after finishing my degree, I went for a PhD to the Max Planck Institute for Psychiatry in Munich, where I did my PhD in neurobiochemistry. After this, I moved then to London, to King's College, at a MRC center we were at the time in London, where I worked for 6 years as a postdoc and later as a research fellow in developmental neurobiology. This, as I said, was for 6 years in London, and after this, I got a civil servant position in France. Maybe you might not be familiar with the system in some countries, like in France and in Spain. The research career, there's a civil servant career system, if you want. In France, there are two actually. This is the CNRS and the INSERM. And so I got one of these positions at CNRS for a year. This was in Paris, in the center south of Paris, in the CNRS center, also working on neurobiology. But I have to say, when I took this position, I was already psychologically lost to research. So this is why I then stopped and resigned from my position after a year, and I moved to Spain, where I am till today. So I left research in 2001, and from there on, I worked in research management, etc. So I stopped doing active research myself more than 25 years ago, or 25 years ago. So I started initially in this new center, the Spanish National Cancer Research Center, which was just being opened and built essentially when I moved there. And there I led the project office, which is essentially the fundraising office and the tech transfer office until 2007. This was actually not six. Sorry. No, it was 2006. Sorry, is correct. And then in between, as this was very much related to my work, what I was doing in Spain in fundraising, we wanted and got in the end a lot of money from the European Union funding streams. So this was very interesting to go for this option, which is called Seconded National Expert, END, which you can do at the European Commission. So I was there for a year and a bit in the Directorate Health in Brussels, and explicitly in the Cancer Research Unit. So just to expand a little bit on this END thing, which you may not have heard of before. First of all, the European Commission, you have heard of for sure. So the EC is the executive branch, the government, if you want, of the European Union, responsible for legislation, implementing EU policies, etc. Mainly based in Brussels. And there's the option, which is what I mentioned before, to do these seconded national experts, or END, which is the French abbreviation. So this is mainly meant for public administration or civil servant from member states. So you have to apply, and then you can go for a specific amount of time. I think this is up to 3 years or so, at least at the time. You can go to work at the European Commission or the European Parliament. And the idea is so that you go there, and so to learn the inside functioning of the EU machinery, if you want. And then you bring back this knowledge, and nothing confidential, obviously, but to gain a better understanding how the wheels turn in Brussels, and so to help institutions to better understand how the entire thing works. So this is the idea of this END thing. And so this is what I did as a Spanish END at the time. So I was sent from Spain. And so this was in 2005 I started. This was DG Research, or the EC then, so 20 years ago. So I was at DG Research. The focus was on, as it says, research and technological development. And at the time, the DG Research and other DGs, but especially DG Research, this was a DG that was in charge of both policy definition and implementation. So they defined the areas of interest for themselves, and they made work programs, calls, etc., organized evaluations, They monitored the grants they awarded. So this was all done by the same Directorate General and the different units within this DG. So this was a mixed DG, if you want, both developing policies and implementing them. And so they had fewer agencies and less delegation to other units in the European Commission. This has changed since, and so this is how this looks today. This is much different, and I think it's less interesting today. So this is also the name has changed. Today it's called DG Research and Innovation. Just to point out here the main changes. So now, these days, DG Research and Innovation is only in charge directly of policy and strategy, but not any longer in charge of the implementation of their own policies and strategies. This has now been, they have since then, so in the last 20 years, they have established a lot of agencies in Brussels. I think most of them at least. Here, the Research Executive Agency and the other abbreviations, I don't even know, so what they mean. But there's quite a number of agencies, and these agencies, they are in charge of implementing the policies developed by the EC. So if you today would go to the EC and would do what I did 20 years ago, you would not work directly with research projects funded by the EC. This is now handled by the agency. So this is a huge change and huge difference compared to 20 years ago, just for you to understand. So, but this for me, this was a great experience. So when I was there for a year and a half or so, this was a great environment, very international, very professional, very interesting, also due to this mixed work of doing policy and implementation. And this in my career from then onwards also, this really helped me a lot in understanding how this works. So this was really for my career very valuable. Okay, let's go back to the table here. So then I went back after Brussels to to Spain, but to a different center, to Barcelona this time, in which is still called CRG, Center for Genomic Regulation, directly on the beach, I have to say, very good spot. And there I was for a year, and then I moved back to Madrid to the special Spanish National Cardiovascular Research Center, where I also headed the project office and the tech transfer office. Then I did also an MBA, an executive in between. And then with a colleague and friend of mine, we founded this company in 2010, which was focusing on drug discovery for rare cancers. And then this was a difficult time, as you may remember, it was in plain crisis, economic crisis, which started in 2007. So it was certainly not a good moment to start a company looking for investment, so not a good timing, I have to say. So this did not proceed far enough to continue really. So this is when I joined then in 2012 EVI, where I'm still today. I started as head of business development, and now I'm director of external affairs. So, and the rest of my talk, I will explain to you what EVI is doing and what EVI is doing mainly. So EVI. So we were established end of the 90s. We're a non-profit organization. We very often call ourselves a product development partnership. So our interest is to support the development of vaccines for global health. So our heart lies with low and middle income countries. So we were established in 98 as European Malaria Vaccine Initiative. So we focused only on malaria vaccines, but since 2009 or so, we have broadened our scope also to other diseases of LMIC interest. And what we do, so we don't have any labs. We are, if you want, a virtual institution. So we always work in partnership. So we essentially further develop the vaccines of other people, if you want. So I explained this a little bit later in a couple of slides for you to understand. So we establish partnerships with other research institutions, And as a consortium, we advance and move forward the development of vaccines that are in our interest, in our mission's interest. And our mission and our vision is just put here below. And in the context of these research projects, we also, whenever possible, we try to integrate capacity strengthening activities to strengthen the clinical research capacities in the partner institutions in Africa or elsewhere in LMIC countries, and also are engaged, which is also part of my work in my position, to support and implement advocacy. So if you want lobbying, sounds a little bit ugly, lobbying, but that's the same in the end, advocacy for global health research. So this is what EBI does. And just to explain why this is still necessary, so I prepared the slide. These are figures from before COVID. So this is not even with COVID included. These numbers would be a couple of million higher, at least in peak COVID times. So as you know, so before COVID, there were still like 8 million people dying every year of infectious diseases, and most of them in LMICs, and even worse, most of these are children below 5 years of age. So this is really terrible. And just listed a few of the main killers: lower respiratory infections, diarrheal diseases, TB, HIV, malaria. No, they are the main killers. And just for malaria, every minute, if you calculate this, a child dies. So while I'm speaking, 20 children have died in this time from malaria. So this is still a terrible burden here. And so this is, and many of these diseases are diseases which have no commercial interest for companies. No, GSK will not work on a vaccine for leishmaniasis or something. No, this is of no interest. Many of these diseases are no interest. And this is where institutions like EVI step in. We try to support the development of products for indications and regions where there's no commercial incentive for at least in the earlier stages or mid-clinical stages to get these products developed. So, and as all of us know, vaccines are a fantastic invention, or if they work, they're extremely cost effective. You don't get sick, so you prevent and do not treat. And usually they can be extremely cheap and effective. So vaccines is really it's great stuff, vaccines. But as I said, there's still an enormous need for many and improved vaccines, and this is what we are supporting at EBI. So, and as I said, we don't have own laboratories. So what do we do? What do we have? So we have a team of experienced staff, mainly obviously from staff that is experienced in research and clinical research related to vaccines. So we do, for example, so what we try to do is to move vaccine candidates that are in our interest, to move them from preclinical research, from preclinical stage into the clinic. So this is what we do. What you can call this translational R&D management. And we also support preclinical development. We do clinical trial support. We sponsor clinical trials. We can manage clinical trials. We try to combine this with implementation research and also integrate social sciences. And as mentioned before, wherever we can, we try to combine this with training and capacity building. And all this is underpinned by project and network management and resources, because we are completely dependent on external funding. We are not the Wellcome Trust. We don't sit on billions of euros. So we always have to look for the money we need for our projects. And this is part of my work, my responsibility to look for the money and get it. So just maybe this explains better how we work, but it took me a long time, even while at EBI, to understand how we actually work. Maybe this makes it a little bit clearer. So EBI here, we are here at the top left. As I said, we have our mission, our interest. Let's say we are interested in a vaccine for leishmaniasis, let's say. No? And then we look we make a landscape analysis and look for institutions that may be developing a vaccine for this particular disease. And then, if we're lucky, we find one here, which is the research group below, which can be from academia or can also be an industry partner, whatever. No? But usually this is either a small industry company, like a small biotech, or academia. So and very often these people have no experience or knowledge on how to move vaccine candidates from the preclinic to clinical stages. No, this is what we can offer as an added value. So if we find a partner like this who works on a vaccine of interest for us at an interesting stage, we contact these researchers and say, let's meet, let's discuss. And we propose them, let's make a partnership to develop your product jointly further. If we come to an agreement, this usually requires additional partners for whatever. This could be partners for animal model development, for animal testing, for data management, for different kind of assays. So we try to look for other partners that we need to complete our consortium, if you want, our partnership. Then once this is established, we jointly, again, as a partnership, we try to find the money, which often we do, not always, unfortunately. So, and once we have the money secured, then these projects are jointly implemented, and they can range from late preclinical stages to, in our case, phase 2 clinical trials, for example. So we cover quite a range in vaccine development. So this is how we function all the time. And then if you're lucky, this repeats. I mean, and usually it doesn't work with a single clinical trial, phase 1, and then you move forward. Very often you have to repeat a phase 1 with a different formulation, with a different platform. So this is a little bit reiterative at the beginning very often, but this is how we work. No? So we partner, we look for funding, implement the projects, and thereby we move these projects further. This is just to give you an idea what we are working on at this moment. So these are the pathogens we're working on. We have a huge portfolio of different malaria vaccines. Malaria, so Plasmodium is a very complicated pathogen, different stages of development, so very complicated, very difficult. So we have multiple vaccines for different and combinations and platforms for different pathogen stages. And then there are different pathogens. There's falciparum, there's vivax, very complicated. In different stages, we work on leishmaniasis. We work on some diarrheal diseases, Shigella and ETEC, on a vaccine for Nipah, a very nasty virus, and also on influenza vaccines. This is our current portfolio. This is our partner network. So we are working or have worked with more than 200 partners from the entire globe. Most of our partners are from Europe, because this is where most of our technologies come from. But then the biggest partner network we have in Africa. So we have a huge African network in sub-Saharan Africa. And we have worked to date with more than 50 countries from across the globe. And these are, as I said, we are depending on external funding entirely. These are our current funders. So there's a lot of different funders, a lot of different European funding streams, but also funding from some national governments, from some other foundations, from industry, from Gavi, and other foundations. So it's quite a mixture. But this is important to diversify, to reduce risks, and this is how we function with these different funders. And I think this is the end of my presentation. I don't know how we are doing time-wise. Was I too fast? I'm okay time-wise.
It's very okay time-wise. Thank you very much. So
Okay.
I want to remind people that they can ask questions in the Q&A. So please, if you have any questions for Stefan, go ahead. In the meantime, I might ask you, I do have a couple of questions for you. Maybe for so you explained how basically to become to collaborate with ABI, but most of the time, then I have the feeling that you are the one contacting, directing the people you want to collaborate with. So if there was someone in our audience that would be interested to show the results of his vaccine, what should he do in order to get in contact with you guys and have a chance to collaborate?
It's just very easy. We do this very often, or people just send us an email. Or do you see my email address here? It's just send an email and say we are working on this and that. If we have time to discuss this, and this we do all the time, to discuss with different institutions and partners and interested parties to see if there's any possibility to collaborate. And from these discussions, very often things start, no? And sometimes it's not relevant for us or too early, too late or whatever, no? But this is how we function, no? I mean, people come to us. I reach out to people I've never met before, no, for the same reason. I ask, I've heard that you're working in this and that, but this is how we initiate our partnerships. Now, this we work a lot, we repeat a lot with partners we've worked for a long time, but there's always new partners joining, and they come like this, no, they come out of the blue very often. So that's easy. Just reach out.
Thank you. Then, for people interested, please reach out. You have the email here. And then I have another question for you from the chat. So, what does the advocacy function of your work look like?
This is so this has different levels if you want. No, this can be on national level. So we work a lot. And also this we do in partnership with other institutions. So EBI is, as I said, a PDP, a product development partnership, but there are many others similar to EBI that have a different focus on drugs, on diagnostics, on different diseases. So we do this very often together because we have a very similar mission. So we work then, for example, on member state level. So we try to lobby, as I said, with national governments to repeat funding calls for our types of institution, to publish calls to support product development, vaccine development for products targeting diseases endemic in LMICs. We do this a lot on, so we are planning an event, for example, next year in Berlin with member of parliaments, to 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 get champions, on parliamentary level, that in the parliament then push the government to provide funding lines in their government in their annual budget for supporting vaccine research for LMICs, for example. This we do on different member state levels. The same we do at European Commission level. Now we work sometimes with member of European Parliament. We meet with European Commission staff. This is what we do. We publish jointly pieces, on highlighting what we do and how important this is, to get support from policy and decision makers. So which is not easy these days because I have to say COVID was terrible, but also COVID was great, because this really showed how vaccines work, what they can do. This has really gave a big push to, at least for vaccines and I guess drugs either, showing that these things really work. And governments spend a lot more money on this kind of research, and this is what we do. We try to show the importance and relevance of what we do and try to maintain these funding lines, which is not easy these days, with wars going on, all these things. So this is something that requires all the time pushing the ministries and governments.
Thank you. It's a very, very interesting work indeed. So I suggest that we pause the question for the moment. I still have a few, but let's keep them for the end of the webinar, for the Q&A. So please, Romain, share your presentation. So Romain Guyon, Romain did a master in biomedical engineering in INSA in Lyon, and then began a PhD in clinical medicine at Oxford, together with Professor Anita Milicic, Stride, and Adrian Hill. And his research is focused on microfluidics continuous process for liquid core PLGA shell microcapsule that co-delivers a priming vaccine and a release booster. So now he's a postdoc, always in Milicic group at the Jenner Institute. Yeah, we're very happy to hear about your presentation. So please, Romain, the floor is yours.
Perfect. Can you hear me well and see the slides?
Yeah, I see and I hear.
Amazing. Thanks so much. Thanks so much for the invitation. I'm very glad to talk about our research. So today I'm just going to cover what we've recently published about the malaria vaccine and also what we are working on right now on the rabies vaccine. So my work is about microcapsules for single shot vaccination. And the concept is very simple. It's to replace your classic prime boost vaccination, which involves multiple injection, by one single shot that contains both your soluble vaccine, the prime dose, and booster doses, but encapsulated inside microcapsules and co-injected. And these microcapsules, they can sit inside your body, they degrade over time, and they release the booster dose after a certain time without the need for a second injection. So to do that, what we've developed during my PhD was this core shell microcapsules. They are 60 microns in diameter, so they can be injectable. They can pass through a 25G needle, which is the standard accepted for human intramuscular injection. And they have a core shell architecture. So they're made of a liquid aqueous core that contain your vaccine, your adjuvant, your excipients, whatever you want to put inside, surrounded by a solid shell of PLGA, which is polylactic glycolic acid, a biodegradable polymer, which is biocompatible, FDA approved. And this polymer can degrade, and the degradation rate will tune the delay of delivery of these microcapsules. So first, let's talk about production. To produce these microcapsules, we developed a microfluidic platform. So it's based on the classic double emulsification method, where we use a microfluidic chip and inject fluids inside. And because of the properties of the channels, we are able to generate double emulsions. So these are the video that you should be able to see on the lower left part of the screen, which are live, like real-time videos of the production of these microcapsules, where drops of vaccines are flown inside a fluid made of the solvent and the polymer, and each drop of vaccine is surrounded by bigger drops of solvent and polymer, and we can then collect these double emulsions, extract the solvent by solvent extraction, which will harden the polymer around the core and form our microcapsules. So you can see how they look like after production. It's on the lower right part with a fluorescence payload inside to represent the vaccine. And one of the big advantage of this microfluidic platform is that you can really have a uniform population of microcapsules with a repeatable dose loading and a quite high throughput, because each microfluidic chip is able to produce capsules at about like 200 microcapsules per second. So we then tested these microcapsules first in vitro. So we put them at 37 degrees in PBS to simulate the body conditions and looked at the release of a fluorescent payload. So these are the kinetic curves that you can see on the left part, where we tested different formulation of polymer, so different formulation of the shell, and so at which rate the payload, the vaccine inside, was released. And you can see that by changing the formulation of the polymer, we were able to achieve between 2 weeks to up to 5 months of delay in vitro. And the way it worked is quite visible on the scanning electron microscopy images that we've done on the right part of the screen, where you can see right after production some microcapsules that either like the intact ones or section of it, where you can see that the shell is intact, they're spherical, they seem that they don't have any degradation signs. But then after a certain time put in vitro at 37 degrees, the polymer went through hydrolytic degradation, and some pores were formed inside the shell. So a full network of pores that you can see on the right-hand side of the screen. And these pores, they connect to each other, and at some point they form a path between the inside of the capsule and the outside. And that's what triggers the release of these microcapsules. That's how they work. We then decided to do some proof of concept in vivo using mice and the malaria vaccine that was developed in the Jenner Institute, so R21, which is a VLP, adjuvanted with SMNP. And in this in vivo proof of concept study, we had 3 groups of interests. So first, you standard prime boost, which is in gray, where mice received 2 injections of vaccine at week 0 and week 2, with every time like a full dose of R21 and adjuvant. We had a control group, which was our prime only, which contained the 2 doses of vaccine and adjuvant in 1 single injection. And then our group of interest, the prime and microcapsules that we received in 1 single injection, both the soluble dose outside and 1 entire encapsulated dose inside microcapsules that were supposed to deliver after 2 weeks in vivo. And we followed the immunogenicity, so the specific antibody response against the R21 malaria vaccine. And as you can see on the graph on the left part, for the first 2 weeks, the response was similar, which makes sense. They were both exposed to the vaccine coming from the prime. And then the prime-only group mice immune response plateaued, while the prime boost continued to rise, which came from the booster that was given at week 2. But you can also see that from the prime and microcapsule group, you also have this rise of antibody titers, which confirms the fact that these capsules were able to produce this booster effect. We also looked at the efficacy in vivo of these microcapsules by challenging these mice at different time points with malaria and looked at the sterile protection coming from the different regimens. And you can see here the results of the logistic model that we run with the different time points of challenge. And as expected, the prime-only regimen didn't protect much the mice, and also the protection decreases with time. By week 8, it's below 20% of protection. For the prime boost, it's quite steady around 70% of protection. And for our group of interest, again, you see first that it's much higher than the prime-only group, and it almost reached the level of protection given by a prime boost regimen by week 11, which was very encouraging. So this is a little summary of all the results that we've had with the malaria vaccine, and you can see the detail of the study in recent paper that we published in Science Translational Medicine, and you just have to scan the QR code if you want to access the study. So following this proof of concept, we are now focusing on 2 things regarding the technology. First, scaling up, and then identifying different vaccine targets or other therapeutic targets that could benefit from the ability of delayed delivery. Because this seminar is about vaccines, I'm not going to expand too much about scaling up, but right now we are basically trying to increase the throughput of production of the microcapsules by parallelizing the chips together, by trying to play with the geometry of each microcapsule so we could just push more vaccine inside. And also by trying to standardizing the production of the microfluidic chip so we can have like some form of a supply chain in place for further production. But what I'm going to talk about is our new focus right now, which is the rabies post-exposure prophylaxis using an inactivated virus vaccine, which is commercially known as Verorab or Rabipur. And right now, when you get exposed to rabies, you get a multiple injection regimen, which is usually given in 3 to 5 injections. So one of the mostly used across the world is the SN protocol, and it involves 3 injections at day 0, day 3, and day 7, and then 2 later ones sometimes at day 14 and day 28, with the objective of producing neutralizing titer antibody by day 14 to protect you against the potential exposure of rabies that you just got. The problem with this regimen is because there is multiple injection, it suffers quite low compliance. And there was a study that was done in an hospital in New Delhi in 22, where about 40% of the participants dropped out after the first dose, after day 0, and they didn't get even their day 3 or day 7. So what we could do would be to, again, try to develop this single shot approach where we would have both the prime injection and the 2 booster dose inside microcapsules and be able to deliver this within these first 7 days to improve massively the compliance of this regimen. The main issue is that we tried to test this with the rabies vaccine with our normal microcapsules. So here, what you can see are IVC images. So they are live experiments where we added a fluorophore tag to the rabies vaccine to make it fluorescent. And we either like injected it as a bolus into the leg of a mice or of multiple mice, or we encapsulated this fluorescent rabies vaccine inside the normal microcapsules that are presented in the past using our fastest formulation that we had access to. And when we looked at the evolution of the fluorescent signal inside the leg of the mice, which is an indication of the release of the vaccine, we could see, so you can see it on the images, but also on the quantitative result on the right part, we could see that, yes, the bolus gets cleared very quickly. So you get exposed to the vaccine very quickly. That makes sense. It's in a soluble form. But once you expose it to once you encapsulate this vaccine inside our microcapsules, you don't have any release that happened before day 12, day 14, which means that it's way too late for this post-exposure application. So we have to improve the technology. And that's what we did recently. We've evolved from the microcapsules to what we call the quick release microcapsules by adding a tiny change during the production, which is the addition of a doping agent inside the shell alongside the polymer. And you can see here a side-by-side comparison between our standard technology, the microcapsule one on the left part, where you can see on the SEM image that the shell is completely intact right after production. But when you add this doping agent, what happens is that you create artificial pores from the moment of production inside the shell, which is on the right part of the screen. And you can also see it on the microscopy images. You really see that the morphology changed. And these artificial pores, they are as if you are pre-degraded the capsules before injection. So they're going to release faster and therefore might match the release window that we're trying to aim for the rabies vaccination. So we tested that in vivo kinetically again. And as you can see, so this time I added the QR microcapsule group, so the quick release group. You can see that the signal in mice decreased by day 4, and by day 8 was almost gone. And quantitatively, you can really see that this release really happened within this first 7 days window, which would be quite relevant for the rabies post-exposure prophylaxis. One of the advantages of this new version of the technology is that you can also tune the delay of release by playing with this doping agent. So here I showed you like 3 different parameters with confocal images. So this time it's a little bit confusing, but the rabies vaccine is labeled in blue and the shell, so the polymer, is in brown, yellow, or purple, pink. And what you can see is like by changing the molecular weight of the doping agent that we put inside the shell, you have bigger or smaller pores. And then quantitatively in terms of release kinetics in vivo, you release faster your vaccine from these capsules when you have bigger pores than when you have smaller pores. You can even extend a little bit further the release by changing the thickness of the shell, which is the pink formulation. So we did lots of experiments in the past 2 months, actually, in vivo, kinetically, to try to identify, to create like a full library of these different formulation, playing with the different parameters. And we use that to simulate the perfect, by simulation, to identify the perfect formulation that would suit our application for the rabies PEP. And what we found, which is going to be my last slide, is we identified this gold quick release microcapsule formulation. So on the right part of the screen, the graph here is the results of the prediction coming from our in vivo data in terms of vaccine exposure that would happen if you get either your standard regimen in gray, so your 3 injection given at day 0, 3, and 7, where you get your 3 doses of exposure, or if you get in 1 single injection, a soluble dose, and 2 doses of the vaccine given in this specific formulation that we identified of quick release microcapsules. And you see in terms of exposure profile that you get something that is very close to the standard that exists. But this time, instead of having 3 injection, you just have everything in a one and done approach. So this is all kinetic and very engineering. We are now trying to validate this immunogenetically, and it's running right now. So we've already shown that the vaccine is stable inside these microcapsules, these new QR microcapsules for 7 days. And we are now running the efficacy study, which will try to assess the level of neutralizing antibody titers after this one and done approach to see if we can completely replace the standard injections. This is the end of my presentation. So I just want to thank a lot Adam, Lee, Soren, and Anita from the Genome Institute, and Luca and Eleanor from the IBME that were involved in the study. And I'm very happy to answer any questions. You can also contact me. Just put my email. Thanks.
Thank you so much, Romain. It's very, very interesting. So, at the beginning of my PhD, I was working on something similar, so I would have a lot of questions. But
Okay.
we will leave the floor a bit to the Q&A. So, just a reminder, please write all your questions on the Q&A. We have some time to go over the questions. So, first of all, I have a question from Sida Wang. How are the porous QR microcaps achieved?
So basically, we add this polymer, this doping agent, during the production phase. So if I just go back here, during the production here. So these are, before they turn into solid shell microcapsules, they are double emulsions. So it's a liquid core with the vaccine, a liquid solvent, and the PLGA polymer. But this time, there's the PLGA polymer and the doping agent. And because the doping agent is going to like diffuse out during the extraction of the solvent, it will leave some void, some pores, and that's what's going to create the artificial pores without degradation.
Great. So I have another question. So are they freeze dried? How do you prevent otherwise that to have the release during just the stability period?
So it's a very good point. So for the normal microcapsules, we just keep them in the fridge in suspension because the degradation at 4 degrees, and we measured it for at least 2 months, the degradation at 4 degrees is too slow to release the vaccine. Obviously, as soon as you add pause, you accelerate the release. So it's fine right now for a week, this which I've been testing so far in the fridge at 4 degrees. We're also trying to dry these microcapsules, so shelf dry them for now, but freeze drying will be the next step. And this will be part also of what I've laid out in my development roadmap, what I call the process development. So identifying all these different aspects that would be required to bring this technology towards clinic. So the storage is a big one, but we are trying to focus first on what's going to be the application before trying to think how are we going to store them. But freeze drying will be one option. Shelf drying is another one. There are recent work also that's been done in embedding vaccines in general, but it works also for PLG capsules inside sugar matrices, which slow down the degradation a lot, and that could be a way to store them.
Interesting, thank you. So I have another question from the Q&A. So Dr. Michael Francis, very nice technology and encouraging data. But if someone has an adverse response to the primary dose, how do you prevent the delayed booster?
It's a very good question. It comes up often. It's a drawback of this kind of technologies. You gain in compliance, you lose in the control in case of adverse effects. So at the end of the day, it has to be a balance between risk and benefits, as usual with therapeutic interventions in general. Because these capsules, so each capsules only contain a fraction of the entire dose. So to give you an example, when I inject this to mice, I inject about 100,000 microcapsules. So you cannot like extract them in some way. So it has to be balanced against what adverse effects you may have. For the rabies vaccine, that's what's cool is not necessarily cool, but like what's interesting is that the disease itself is deadly. Like if you develop rabies, it's very bad. So it tends to be a very good case where this would be very useful. Because the worst that can happen could be allergic reactions. And the way to prevent that could also be to give in one single visit to the healthcare a first injection of your bolus, then wait for a little bit to see if there's immediate reactions, and then give a second injection, but on the same day. of the capsules. So that way you can control at least the immediate reactions. It defeats the purpose of like a single injection, but at least you gain in terms of logistics because people don't have to come back. So there are like different ways to adapt this, but ultimately it's something to weigh against the benefits.
Thank you. So I have a question for Stefan now. Well, we will have a lot of questions. So mine is a very maybe naive questions, but at which point during your career you realized you were very good at raising money?
Sorry, at which point of my career, what?
You realized you were very good at raising money, that you decided to switch from research to go in that direction.
Well, I didn't know beforehand, but I definitely, I think you have to like writing, no? Because what a lot of the work I do is writing, no? I mean, not only emails, but also I also do write certain parts of proposals and what all kinds of things, no? So if you hate writing, this is probably not something you should get into. But then I didn't know before, no? But I turned out to be useful, let's say. So you will not know until you try, no? So.
Yeah, thank you. So you had also a question for Romain, so I will just let you ask it directly.
Yeah, it came to my mind if you can use this type of coating also for oral vaccines, so they go through the stomach without, like for live attenuated vaccines. Could you coat them in? Is it also suitable for this, or is this completely wacky?
It definitely could work for oral vaccines too and protect them. That actually has been done with similar type of technology. The only thing I would say is one of the key aspects of this is that it's injectable because we always thought, can we do something that can be easily rolled out on the field for low and middle income countries? But it comes with so we were trying to make them as small as possible so they could pass through a needle. For oral vaccine, then I would say you could just design an implant that you could swallow as long as it's not too big, and it would like makes the production faster than the process right now. So yes, in fact they could work, but to be too fully honest, there must be other technologies that would have the same principle. So coating them, but that would be bigger, so you have like a bigger encapsulation efficiency.
Thank you. So I have another couple of questions for you, Romain. So
Mhm.
I have a question from Chen Zamir. What happens to the antibodies titer in the long term?
I'm laughing
Months, here?
yeah, I saw the question. I saw the question. Um, we just started this when I started my PhD, and the immune studies were happened like 1 or 2 years ago. So the first one. So we don't have like long term data, and it's obviously difficult to do it with mice. The ultimate goal is to have like to keep the antibodies longer for sure, but we have zero data right now to assess that. So we're right now purely trying to focus on like these short boosters and be able to replace the booster vaccination without thinking yet about the extending the half-life of your circulating antibodies or things like that.
Thank you. So I have a general recurring question here that is regarding the types of antigens or adjuvants that you can encapsulate and what are the limitations.
Anything that is soluble in the aqueous buffer we use, which is very standard, phosphate buffer. There's no secret here. So anything that is soluble is fine. The limitations are the stability. And that's why we add excipients. And each vaccine at the end requires some optimization to make sure that they will sustain the condition of the body for the time you want it to be delayed before delivery. So it's more about thermostability of the vaccine. So if you think about mRNA right now, which notoriously is not very stable at 37 degrees for sure, that would be one of the limitations. It could be solved if there is improvement on the formulation part of the vaccine itself. But right now, the technology is working quite well for any protein-based or inactivated virus vaccines.
Thank you. So another question for Stefan. Um, So this is generally a question that I ask with people that have an MBA. So how much did it help you to have it, to build your career? And could you work while doing it, or you dedicated a full year to your MBA studies?
Yeah, so this was an executive MBA, which means usually that you do this in parallel to working. So this was like Friday, Saturday, and Thursday. I think it's a 2 or 3 nights a week for a year. And then you have to do all kinds of homework and whatever. So it's quite intensive, no? But for me, I think it was very interesting and useful. I mean, this is obviously you do all kinds of things, strategy, marketing, accounting, all kinds of things, no? Everything is a little bit superficial naturally because you can't repeat uh, full full 4 or 5 year career in condense this in 3 days a week for a year. But for me, this was also sufficient because I don't want to become an accountant or something. You know? So for me to get a top level view on many of these things, for me this helps a lot. The thinking, you know, how to think, what to keep in mind, how to plan, how to structure things. And for me, this was extremely useful. You know? So I would if someone thinks in going outside research and in these kind of works, you know, professions, I think this is very helpful. You know, if you can, so I can only recommend this. You know, this in my case was definitely a help.
Thank you.
Mm.
So I will go back to Romain. So we have a question. How would you expect your quick release microcapsule to shape the general magnitude of the vaccine immune response?
So it's quite interesting, and that's actually what my PhD student that I co-supervise with Anita and Eleanor, so Lee, works on. There are recent studies that were done in the US that show that continuous exposure, which is roughly what you get from these QR microcapsules, might actually improve the immune response, both in specificity and in magnitude. So we actually, instead of like inducing tolerance as what was thought in the past, experimentally they saw that there was actually a higher immune response because of this repeated injection that they performed in monkeys, in mice. So we're thinking that that might actually be at least the same, potentially better, as long as we keep the vaccine stable, of course. So we're quite excited to do some mechanistic studies too, and that's what Lee Sims is doing. Yeah.
So I suggest we go with one last question and
Mhm.
then we can close here. So Nicolo asked, or rather than PLGA and various formulations of it have already been similar polymers for the same purpose tested. How has been achieved the decision of using PLGA at the end?
Uh, Choosing PLGA was because it was commercially available, plus it's FDA approved, already used for lots of drug delivery systems for hormones. So it's already like if you're bringing a new technology, it's much easier if you use elements that are already used and already safe. So that was the main reason for PLGA. It's also very well characterized by people much more expert than me in chemistry in terms of degradation rates. So I could use this to actually tune the microcapsules release. There are some work that is done with other type of polymers. So PLG is a polyester. They use other things like polyanhydrides, but it's not yet either approved necessarily, or it's still like in very early developments. So yes, it could be usable, but the reason why we use PLG was mostly because it's been proven that it works well to deliver for human use drugs, at least.
Well, thank you very much. I suggest we're already over time, so I suggest we finish here.
Yeah.
So thank you very much for the presentation and also for staying a little bit longer for the Q&A. This was very interesting, and we've seen from also the questions that were asked in the chat that everyone was very interested from the talk. So I'm sorry for those who didn't have response to the question, but unfortunately time is short. So thank you very much. And for attending. And here I close the last webinar of the year. Thank you, Romain. Thank you, Stefan. It was a pleasure to
Thank,
having you here.
Yes. Thank you very much, Allegra.
thanks, thanks so much,
Thank
Allegra.
you.
It was a pleasure.
Thank
Okay.
you. Bye-bye.
Bye-bye.
Bye.
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 Bye.